In this video we will discuss about the structure of antibody. Good morning. Welcome to YouTube channel, Bioconcept, Learn with Concept. As we already know that there are two types of immunity in our body. First one is innate and second one is adaptive, and this adaptive immune response is further classified into two types, the humoral immune response and cell-mediated immune response. In humoral immune response, which is also known as antibody-mediated immune response, this response is carried out by antibody and body fluid. This antibody is secreted by differentiated plasma cells. If we talk about general properties of antibody, then antibody is made by B cell and plasma cell. It's a globulin protein. It's a glycoprotein in nature, which is made by carbohydrate and protein molecules. Antibody can be membrane-bound and secretory. Membrane-bound antibody is found in surface of B cell and function as B cell receptor. This may be antibody M or D. Secretory antibody is found in body fluid, blood or lymph. These are G, A and E. If we talk about the structure of antibodies, then it's Y or D shapes in appearance. Antibodies are complex structure of 4 polypeptide chain, made by 2 heavy chain and 2 light chain. Each heavy chain are typically 55 kilodalton in weight, and light chain has 25 kilodalton. The heavy chain are longer than light chain. This heavy are in 5 types: alpha, gamma, delta, mu, and epsilon. Heavy chain are made up to 420 to 440 amino acids. 2 heavy chain are held together by 1 to 5 disulfide bonds. Each heavy chain is bound to a light chain by a single disulfide bond, and by non-covalent bonds, such as salt linkages, hydrogen bonds, and hydrophobic bonds to form a heterodimer heavy and light. Similar, non-covalent interactions and isulfide bridges link the 2 identical heavy and light chains to each other, to form a basic 4-chain antibody structure, with 2 heavy chain and 2 light chain. Based on the heavy chains, antibody are classified in 5 classes. For example, IgM contains mu, IgG contains gamma, IgA contains alpha, IgD contains delta, and IgE contains epsilon heavy chains. These heavy chains are structurally and antigenically distinct for each class of immunoglobulin. They differ in their size. Carbohydrate are present as this region, and function as antigens. The light chain, an immunoglobulin molecule has 2 light chains. Each light chain is made up of 220 to 240 amino acids. Light chain is attached to the heavy chain by a disulfide bond. The light chains are structurally and chemically similar in all classes of immunoglobulins. They are of 2 types, kappa and lambda. These 2 types differ in their amino acids present in constant regions. Each immunoglobulin has either 2 kappa or 2 lambda chains, but never both. The kappa and lambda chains are present in human serum in a ratio of 2:1. There are 2 constant region and 2 variable region on immunoglobulin. Each polypeptide chain of an immunoglobulin molecule contains an amino terminal part and a carboxy terminal part. The amino terminal part is called the variable region, and the carboxy terminal part is called the constant region. Both heavy and light chains contain variable and constant regions. These regions are composed of 3-dimensional, folded structures with repeating segments, which are called domains. Each heavy chain consists of 1 variable domain, which is called VH domain, and 3 constant domains, which is called CH domain. IgG and IgA have 3 CH domains, CH1, CH2, and CH3, whereas IgM and IgE have 4 domains, CH1, CH2, CH3, and CH4. Each light chain consists of 1 variable, VL, and 1 constant domain, CL. Constant region, the carboxyl terminal half of the molecule is called the constant, C, region. It consists of 2 basic amino acid sequences. The FC fragment is present in the constant region of heavy chain. The constant region of the heavy chain has many biological functions. It is responsible for activation of the complement, binding to cell surface receptors, placental transfer, and many other biological activities. The constant region of the light chain has no biological function. constant heavy chains of gamma delta and alpha, has a proline-rich region, variable region, the amino terminal half of the light or heavy chain, consisting of 100 to 110 amino acids, is known as variable or V regions variable light, VL in light chains and variable heavy, VH in heavy chains. Variable region is different for each class of immunoglobulin. The variable regions of both light and heavy chains consist of three highly variable regions known as hypervariable regions. This region is called Fab region, and antigens bind with this Fab region. The antigen combining sites, Fab of the antibody molecule that consists of only 5 to 10 amino acids. Each are present in the hypervariable region of both the light and heavy chains. These antigen binding sites are responsible for specific binding of antibodies with antigens. Hypervariable region is also called complementarity determining regions, or CDRs. Fab and FC region. Fab region found in variable site, and FC region is found in constant site. The Fab regions bind to the antigen, and the FC region of the antigen-coupled antibody binds to FC receptors on phagocytic or cytolytic cells, or to immune effector molecules. If we talk about functions of antibody, then antibody main functions are: they prevent attachment of microbes to mucosal surfaces of the host, they reduce virulence of microbes by neutralizing toxins and viruses, they facilitate phagocytosis by opsonization of microbes, they activate complement, leading to complement-mediated activities against microbes.